Good day, ma'am. I am Marian Isguera from 3CRIA2-4 of Pinmaarolo University from College of Criminal Justice Education. This is midterm laboratory examination in forensic chemistry and toxicology entitled as diaceribonucleic acid analysis. The objectives I should meet are explain the importance of DNA analysis and enumerate the steps in DNA analysis. So first is, what is diaceribonucleic acid? Diaceribonucleic acid or DNA is the hereditary material in humans and almost all other organisms. So, yung DNA daw is yung nagiging heredity ng isang tao. So, dito malalaman mo na yung isang tao is related sa family niya or sa mother niya or sa father niya dahil sa may DNA. Next is the origin of DNA. DNA is located in the cell of human in the nucleus, and inside the nucleus is the chromosome and found the DNA. So, yung DNA daw is located sa cell ng human which is in the nucleus. And sa inside ng nucleus, makikita mo dun yung chromosomes, and in chromosomes, dun mo na makikita yung double strip or yung DNA. Next is the composition of DNA. DNA has three types of chemical component. First is the phosphate, a sugar called diacylribose, and four nitrogen bases, adenine, guanine, cysteine, and thymine. And now let's talk about the nature of DNA. DNA molecule consists of two strands which are connected together by hydrogen bonds and helically twisted. Each steps on one strand consists of nucleotide of purine base which is alternate with that of pyrimidine base. Thus, a strand of DNA molecule is a polymer of four nucleotides, the adenine, guanine, thymine, and cytosine. The two strands join together to form a double helix. Next is how the forensic DNA analysis help in criminal investigation. Forensic DNA analysis is generally used to solve crimes in one of two ways. In case where a suspect is identified, a sample of the DNA persons can be compared to evidence from the crime scene. The results of this comparison may help establish whether the suspect committed the crime. For example, is na-identify na yung personality ng suspect, and para mapatunayan talaga na siya yung suspect or yung nag-commit dun sa crime na yon is kukuha for example ng strand ng hair ng suspect and i-identify kung yung DNA ng suspect is same sa nakuhang evidence sa crime scene. And through that, ma-solve na yung case and ma-find na guilty yung suspect. And now let's talk about the steps in DNA analysis. First is the isolation of DNA. Make sure to isolate the DNA from evidence sample containing of unknown origin sa mga known origin. Next is the processing of DNA. Proceed in examination of the DNA and make a result. And next is the determination of variation in the DNA test results. After examining the DNA, determine the variations or the type of DNA. And next is the comparison and interpretation. Compare the test results from the unknown and known samples to find if it is the same or not. Next is the types of DNA analysis. First is the polymerase chain reaction. This is the copying process. It is a method widely used to rapidly make millions to billions of copies of a specific DNA sample, allowing scientists to take a very small sample of DNA and amplify it to a large enough amount of to study in detail. So, ito naman daw, si polymerase chain reaction, ito yung tinatawag na copying process. So, ang kailangan lang ng mga scientists is konting amount ng, for example, is blood, then kaya na nilang paramihin yon into million or billions of copies of a specific DNA sample. Next is the Y chromosome. The male specific part of the human Y chromosomes is widely used in forensic DNA analysis, particularly in the case where standards autosomal DNA profiling is not informative. A Y chromosomal gene fragment is applied for inferring the biological sex of a crime scene trace donor. So, dito naman yung Y chromosomes is madalas siyang ginagamit sa mga rape cases kasi dito mo madedetermine yung Y chromosomes kasi is para lamang sa mga lalaki or sa lalaki lang nanggagaling to, wala yung babae. So, malalaman mo yung, or matitrace mo yung donor ng semen, for example, sa isang rape case through Y chromosomes. And next is the mitochondrial DNA. It analyzes DNA found in the different parts of the cell called mitochondrion, such as hair shafts, bones, and teeth, that are unnameable to STR and RFLP. For this reason, mitochondrial DNA testing can be very valuable to the investigation of an unsolved case. Dito naman, ginagamit naman yung mitochondrial DNA especially sa mga matagal ng mga case or yung mga hindi na-solve na case. For example is, ma-identify mo pa rin yung DNA ng isang tao through the use of teeth or yung mga bones or hair shafts. Kasi, pag matagal na yung tao is, hindi na mape-preserve ng maayos yung, for example, yung blood and saliva niya. Again, I am Marian Isguera from 3CRI82-4 and this is presented to Dr. Elenita G. Cuenca. And the objectives I met are explain the importance of DNA analysis and enumerate the steps in DNA analysis.